Alright, so today we're looking at topic 2.4, which focuses on proteins, and this comes from the greater topic 2, molecular biology, from your IB biology syllabus. Alright, so in order to understand proteins, we must first understand amino acids, because amino acids are our building blocks. So the first point from your syllabus states that amino acids can be linked together in any sequence, so in any order, to give a variety of possible proteins. So we've already discussed how amino acids are the building blocks for protein formation. Do you remember what the name is that is given to building blocks in molecular biology? Hopefully you said monomers. So remember, our monomers are our building blocks, and when we put monomers together, we get polymers. So in today's case, we're looking at amino acids as our monomer. When we put them together, we get polypeptides or proteins. So proteins are made of long chains of monomers called amino acids, and there are 20 different amino acids. And these amino acids can combine together in different ways, and that gives us a huge range or a huge variety of possible polypeptides. So amino acids share a common basic structure. So we have a central carbon, which is denoted by C, and this central carbon is bound to an amine group. So amine refers to NH2, and that's why we get the name amino acid. So we have an amine group, we have a carboxylic acid group, so that is a C double bonded to an O, single bonded to an OH. We have a hydrogen atom on that central carbon, and we also have a variable side chain on that central carbon. So this variable side chain is denoted by R, and this side chain gives amino acids its unique properties. So the reason why we have 20 different amino acids is because there are 20 different variable side chains that we can put onto that central carbon, and that gives us a new amino acid. So here's an example of a generalized amino acid. So here are those features again. That central carbon, off that central carbon we have an amine group. Off that central carbon as well we have a carboxyl group. Then we also have our hydrogen atom up on the top, and then we have our variable side chain denoted by R. So this is what we call a generalized amino acid. We can replace this R with a number of things, and that will give us a new amino acid. And so there are 20 different amino acids, which means there are 20 different variable side chains. You don't have to necessarily remember all the 20 different amino acids, but you just need to remember this generalized structure. That being said, I am now going to show you a couple of different examples of some amino acids. So the most simple amino acid we can have is if we replace this R with a H. And that gives us the amino acid called glycine, or as is written in shorthand, GLY. So by replacing the variable region with a H, we get this amino acid glycine. We can replace that variable region with a CH3, and that will give us the amino acid alanine. We can put a number of different things. We can even pop some rings in there. This is a bit of a funky one, and this would be phenylalanine. So this too is an amino acid where that R, so that variable region, we have this replacing it, and that's what gives us this unique amino acid. So we need to be able to draw an amino acid. We've gone through drawing glucose, saturated fatty acids. Now we need to learn how to draw an amino acid, and this comes from a syllabus point from topic 2.1. So shortly I will show a video of how to draw an amino acid. Try and follow along at home using this checklist. So we need a central carbon, we need a carboxyl off one side, we need an amine off the other side, we need a variable region, and we need that hydrogen off our central carbon as well. So here's that video. Try and see if you can follow along at home. So there it is, our generalized amino acid. You can see we've got our central carbon, got our carboxyl, got our amine, we've got our hydrogen, and we've got our variable region. Just thinking back to the fact that carbon forms 4 bonds, so there are 4 different things we need to account for coming off this central carbon. So once again, I sound like a broken record, but those 4 things are our carboxyl group, so a COOH, our hydrogen, our amine group, so NH2, and our R variable region. Now let's have a look at protein structure. So proteins are very, very complex, and so as a result, there are different levels to how we classify protein structure. So there are 4 levels. We'll start by looking at the first level, which is the primary level of structure. So the primary structure of a protein refers to the order of the amino acid sequence. So we've already discussed how amino acids join together in a sequence to make our polypeptide. And so the specific order in which different amino acids join together is what is referred to as our primary structure. So because there are 20 different amino acids and we can combine them in different ways up to different lengths, there are so many different primary structures that exist. So once again, the primary structure of a protein refers to the order of the amino acid sequence. Now we know that our amino acids combine together, and that combination of amino acids, or the coming together of amino acids, is done so by condensation reactions. So hopefully by now you're able to tell me what a condensation reaction is. So I can hear you yelling at the screen. It's where water is produced. It's where water is produced. And you would be correct in saying that. Maybe you weren't yelling at the screen, but that is the correct answer. So reactions in which things or monomers combine together, and one of the byproducts of that combination or of that reaction is water. That is what we refer to as a condensation reaction. And remember, condensation reactions are the main type of anabolic reactions that take place in living organisms. So the linking together of many amino acids is done so by condensation reaction. We know it forms a polypeptide. And we need to be able to draw a molecular diagram to show this condensation reaction. So I will show you an example. We start by drawing our amino acid, which we know how to do now. We're combining 2 amino acids together, so we're going to draw in another one. And what's going to happen is that we know water is going to be produced. So our hydroxyl, or an OH, of the carboxyl is going to combine with a H of the adjacent amino acid. So we've got a hydroxyl coming off this first amino acid, and we've got a H coming off the amine group of the second amino acid. They're going to combine together to form water, so H2O. And then we're going to have our 2 amino acids linked together. That link is called a peptide bond. So here we have the formation of a peptide bond in which the carbon of one amino acid links to the nitrogen of the second amino acid, and that gives us this characteristic NCC backbone. So NCC, NCC. And if we linked many amino acids together, we would get that pattern. So NCC, 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 so on and so forth. And so where we link many amino acids together, we refer to that as a polypeptide because we would have many of these peptide bonds. The second level of structure, or the secondary structure, refers to folding that takes place in our amino acid sequence. So amino acid sequences will usually fold into one of 2 stable configurations, and these configurations are secondary structures. So the amino acid has the option of either folding as an alpha helix, so as a coil or spiral shape, or as folding as pleated sheets. So we call the coil or the spiral shape alpha helix, and we call the folded shape beta pleated sheets. And so this is referred to as the secondary structure. And this folding happens because of hydrogen bonds. So hydrogen bonds will form between our amines and our carboxyl groups of amino acids, and this takes place between non-adjacent amino acids. And what that simply means is that it takes place in amino acids that are not next to each other. So they're some distance away from each other, and as a result of hydrogen bonds forming between non-adjacent amino acids, we get some folding or spiraling of the shape of that polypeptide. So if we have a look here, this one on the left is an example of an alpha helix. And so we see we've got one amino acid here. This part is the amine group because we see that nitrogen. And we've got a hydrogen bond forming between another amino acid at some distance away. So we see here this would be the carboxyl area. We see that by this double bond. So we've got this hydrogen bond occurring between the amine group of one amino acid and the carboxyl group of a non-adjacent amino acid. And that's happening along the length of this polypeptide, which is giving it its spiral form. So that's one way we can get secondary structure. In another way, it might be in a pleated sheet form. So this is simply just because, once again, we have hydrogen bonds forming between the amine group and the carboxyl group of a non-adjacent amino acid. It's just in sort of a different way. So the distance or the number of amino acids between each hydrogen bond is different. So that's simply why this one is pleated and this one is helical. Let's move on to tertiary structure. So the tertiary structure of a protein refers to folding that takes place in a polypeptide due to the variable side chains of amino acids. So the tertiary structure is also looking at some sort of folding, but it is different to the secondary structure. So don't get the secondary and the tertiary structure confused. Secondary structure, which we looked at on the other slide, was folding due to hydrogen bonds between carboxyl and amine groups. So between the backbone of the amino acids, so that carboxyl and that amine from non-adjacent amino acids. Tertiary structure refers to folding as a result of the variable region of amino acids. So in the last slide, we were not considering variable regions. We were just looking at hydrogen bonds between the backbone or between the carboxyl and the amine groups. Now with tertiary structure, we're looking at interactions between our side chains, so that R, or the variable region of amino acids. So tertiary structure, that folding because of those side chains, gives a polypeptide a 3D appearance. So where we looked at secondary structure, we were looking at folding or spiraling. Now with tertiary structure, we're looking at these complex 3D shapes that are going to take place due to a number of different interactions with our variable side chains. So different amino acid sequences will fold into different configurations, and this is because of chemical properties of those variable side chains. So what are those different chemical interactions? Well, amino acids that have some sort of charged side chain, so a polar or an ionic side chain or R region, such amino acids are hydrophilic in nature, so they love water. We know that things that are charged are very attracted to water because water itself is polar. So if we have amino acids that are polar or that are ionic, they're going to love water. And so because such amino acids love water, the way in which they will configure themselves is to face outwards or to face on the outside of the protein and get in as much water as possible or be as close to water as possible. Another type of interaction is if we have amino acids that are non-polar, so things that are without a charge or that are non-polar, they are going to be hydrophobic. So they are going to want to face inwards in the protein, and so they will bend in such a way to avoid water and face inwards because they are hydrophobic. So it's pretty straightforward. Anything that is hydrophilic or anything that loves water, which is our charged and polar substances, they're going to face outwards. And then anything that is non-polar or anything that is uncharged is going to be hydrophobic, and as a result, it's going to shield itself and face inwards in the protein. So that's why we get this bending and this 3D configuration that takes place in our polypeptide. So don't think of proteins as just this linear sequence of amino acids, but remember those chemical interactions that are taking place that's going to cause the polypeptide to bend in different configurations. Another chemical interaction we can consider is the presence of sulfur. So you don't need to understand the chemistry of this too much, but essentially any amino acid that has sulfur in the variable region is going to form a bridge with another amino acid that contains sulfur. And we call these disulfide bridges. So essentially it's like, oh, I have sulfur, you have sulfur, let's pair up. And so a bridge forms between amino acids that have sulfur in their variable regions, and that also contributes to some of the bending that takes place in the tertiary structure. So we can see here that different side chain properties affect the overall shape of the polypeptide chain. And so the shape of a polypeptide is determined by the order of the amino acids. So we can talk about tertiary structures being the folding of a polypeptide due to that variable region, but we also have to consider how the primary structure is going to have flow-on effects to the overall shape that we're going to get in the tertiary structure, because ultimately the order of those amino acids is going to determine the way in which bending takes place. So the amino acid sequence determines the three-dimensional conformation of a protein. Let's look at quaternary structure. So quaternary structure refers to when two or more polypeptides link together. And not all proteins have a quaternary structure, so some proteins are actually fully functional at the tertiary level, so once they've achieved that 3D shape, but we can have different polypeptides linking together, and that gives a quaternary structure. So some proteins do have a quaternary structure, and some examples, or an example of a protein with a quaternary structure is DNA polymerase. And DNA polymerase is an enzyme that is composed of 4 different subunits. So before we move forward, why don't you have a go at trying to define primary, secondary, tertiary, and quaternary structure using a couple of words for each level of structure. Pause this video in order to have some time to do so. All right, another question. Hemoglobin is a protein that possesses more than one polypeptide subunit. Therefore, it has a what structure? Does it have an A, primary, B, tertiary, C, complex, D, quaternary, or E, secondary structure? Pause this video if you need more time to answer the question. So hopefully you said that hemoglobin would possess a quaternary structure, so D. And this is revealed in the question because it says that it contains more than one polypeptide subunit. And so we know that the quaternary structure refers to when we have 2 or more polypeptides linking together. And so hemoglobin here is another example of a protein that has a quaternary structure. Remember, not all proteins do have a quaternary structure. In fact, many proteins are fully functional at the tertiary level. All right, let's have a look at denaturation. So denaturation of proteins occurs by heat or pH. Denaturation refers to a structural loss or a structural change in a protein. And so when we talk about structural change, this means that there's a loss of quaternary or a loss of tertiary or a loss of secondary structure. So we know what these mean now, and so as we look at denaturation, we see that if we have a loss in quaternary, tertiary, or secondary structure, we've got a change in the shape of that protein. Now, denaturation in a protein is usually the result of either heat or pH. So the way a protein folds, so the shape of a protein determines its function. So structure is related to function. And so if we have a loss or a change in the structure of a protein, that means that the protein will lose its function or it will alter its activity. So denaturation of proteins is, like we've mentioned before, caused by 2 key conditions, either temperature or pH. So for example, when we cook an egg, what we're actually doing to that egg is denaturing it, which is why it's no longer runny after it's spent some time on that pan. So we've denatured the proteins in the egg. Now, it's also important to consider what is optimal for a specific protein when we refer to or when we talk about denaturation, because different proteins will have different optimal temperatures and different optimal pHs, and so what would denature one protein might not denature another. So for example, we know in our stomach we've got hydrochloric acid, and it gives our stomach a very acidic, very low pH of around 2 or 3. And so therefore, the enzymes that work in our stomach, such as pepsin and trypsin, they will not denature at a pH of 2 or a pH of 3. But the enzymes that work in our mouth would denature at a pH of 2 or 3, because the enzymes that work in our mouth work at around a neutral pH, because we know that our mouth is closer to being a neutral pH than our stomach is. And so for that reason, we need to consider what is optimal for a specific protein when we are talking about denaturation. So we need to consider the optimum. When it comes to temperature, there's a little bit less variation. So temperatures at which proteins may denature vary a little bit. But for most human proteins, it's going to function at around 37 degrees, because human body temperature is usually around that 37 degrees Celsius. And so most of the proteins that work in our body are going to function optimally at 37 degrees Celsius. If they are exposed to an environment that is hotter than that, then they would denature. We need to understand how proteins are made as well. So your IB syllabus states that amino acid sequence of polypeptides are coded for by genes. So here is a nice little diagram to sort of visually represent how proteins are made. So first we start by looking at a chromosome. So sections of chromosomes are referred to as genes. So this section here would be one gene, this would be another gene, this would be a third gene. So each section of a chromosome is a gene, and each gene will code for a specific protein. So let's say we take gene 1 here in blue. If we zoom in, we can take a DNA strand from that gene. So we know that chromosomes are made of DNA. So as we zoom in, we see this is the DNA sequence. Now, when we transcribe that sequence, we make RNA. So the process of transcription is going from DNA to RNA. And the reason why we need to do this is because DNA cannot leave the nucleus, whereas RNA can leave the nucleus. So when we transcribe this DNA into RNA, our RNA will then leave the nucleus into the cytoplasm. Then at the cytoplasm, it's going to encounter a special organelle called a ribosome. And at the ribosome, this RNA is going to be translated into amino acids. There's more about this when we look at protein synthesis later on in this topic. But as a brief rundown, we've got codons, which are made of 3 bases. And when those bases are split through the ribosome, it puts out an amino acid. So for every 3 bases, or for each codon, we get an amino acid produced. And then these amino acids are joined together to make our polypeptide. So now obviously this was just a very simple rundown of the process of making proteins, but there'll be more about this later in the topic of molecular biology. I believe it's topic 2.7. All right, let's have a little bit of a look at what proteomes are. So a proteome refers to all the proteins that is expressed by an organism. So every individual has a unique proteome, and this is because protein expression, or the proteins made by an individual, depends on that individual's genes. And so that's why each person or each individual has a unique proteome, because every individual has unique genes. So while one's genes does determine their proteome, the number of genes is not equal to the number of proteins that an individual makes. And the number of proteins expressed in an individual is always significantly more than the number of genes that that individual has. And this is for the following reasons. Firstly, a gene sequence can be spliced, which it just refers to cutting sections out of the gene after transcription to make a new gene that will code for a new protein. So we might have a specific gene that codes for the production of one protein. However, if we edit that gene a little bit by splicing and cutting out different sections of it, we end up with a new gene that's going to code for a new protein. And so we see there that this is a contributing factor to why the number of proteins expressed in an individual is actually more than the number of genes that we have. It's because we can do a lot of editing to genes to make that gene produce more than one protein. Another factor that contributes to why the number of proteins that we have is more than the number of genes we have is because polypeptides can also be modified after translation to form a whole new protein. And so we've talked about quaternary structures and how different polypeptides can link together to form a whole new protein. And so we can see there how that would be a reason for why the number of proteins that we have is actually more than the number of genes we have. There are a number of different modifications that can be made to make whole new proteins without necessarily having the same number of genes. And so hopefully we can see here that proteins are very diverse. We can change one amino acid in an amino acid sequence, we've got a whole new protein. We can have splicing take place of genes to make whole new proteins. We can stick different polypeptides together to make whole new proteins. And we can see that proteins are very diverse. So in humans alone, there are millions and millions of proteins. If we think about all the other different living things, think about other animals, think about plants, fungi, bacteria, protists, they all have a diverse number of proteins because every living organism can synthesize many proteins with a wide range of functions. There are billions of proteins in existence, and each protein has a different function. So let's have a look at some different functions. As we look at these functions, we're going to look at some examples because there are some named proteins that are written in your syllabus that you need to have an understanding of as examples of the range of protein functions. So here's a nice little table. If we think about this column here, we're looking at the different protein functions, and then in this right column here, we've got different examples of those. So we start by looking at structure. So proteins can function as structure. And some examples of that are collagen, which is found in animals' skin, nails, hair, bones, and it provides structure. And we can also look at spider silk. So we know that spider silk is a protein that is involved in the formation of spider webs, and it gives those spider webs that nice structure. So we can see here that proteins can function as structure. Proteins can also have a hormonal function, and as an example, we look at insulin, which is a hormone responsible for regulating blood sugar. So when blood sugar levels are high, insulin is the hormone that is responsible for reducing our blood sugar levels. Proteins can also function for immunity, and as a general term, we call the proteins that help in immunity immunoglobulins. Or a more specific term that we can use are our antibodies. So antibodies are immunoglobulins, which are a type of protein, and they function for immunity. We can think about proteins in transport. So we know that when we look at the cell membrane, we have a number of different proteins that span the width of the cell membrane and allow things to go in and out of that cell. And so proteins, particularly membrane proteins, can function for transport. Proteins also function for sensation. So I'm sure you've heard of your rods and cones in your eyes. So rhodopsin is an example of a protein that is found in your eye, and it is a photopigment. So it allows for the sensation or the detection of light. So there is another protein function, sensation. Another one is muscle contraction. So for those who are doing HL biology, you do need to have an understanding of muscle contraction, and actin and myosin are the proteins that are the contractile units of the muscle. So they contract, and so we can see that proteins can also function as units for muscle contraction. And the last protein function that we're going to look at is the function of being an enzyme. So enzymes are biological catalysts, so they speed up the rate of reactions that take place in living things. An example of that is Rubisco, which is an enzyme that speeds up one of the reactions that take place in photosynthesis. So in order to remember these different functions, there's a nice little acronym. or mnemonic, and I'll let you have a go at remembering that. So remember all these different protein functions and be able to give some examples. So the examples that specifically you need to be able to give are collagen, spider silk, insulin, immunoglobulins, rhodopsin, and rubisco. That's all for today's video. Happy studying.